This is a series of 3 short videos that aim to provide an overview of the neural pathways and areas responsible for visual perception and seeing. In particular, we will go over how light information from the physical environment is detected, coded, and then processed by the brain. These videos are meant to supplement and ground your understanding, and I refer you to the lectures for additional information and resources. We start off with this schematic representation of the brain and the neural pathways that form our visual system. This system can be broadly divided into 3 regions: prestriate areas, comprising of the retina, optic nerve, optic chiasm, optic tract, and lateral geniculate nucleus; the striate cortex, and the extrastriate cortex, comprising of areas beyond the striate cortex in the occipital, temporal, and parietal lobes of the brain. As you can see, this division is relative to the striate cortex, or primary visual cortex, or V1, which is a very important area as it is the first to receive and process visual information from the eyes. In these videos, these divisions will be discussed in detail, particularly their organisation and function. In the first video, we'll be talking about areas that comprise the prestriate visual system, which is the first stage of seeing. The process of vision starts off with the distal stimulus, which is the external world, and the image of the external world, which is contained within the visual field. The visual field represents the entire visual scene that is visible to both eyes. This visual field can be nominally divided down the middle into left and right visual fields, and also into upper and lower halves. This forms 4 visual field quadrants with substantial left and right eye overlap. Light from the visual field projects to the eyes, and the optics of the eye work to focus light onto the retina located at the back of the eye. The visual pathway begins at the retina. Because of optical inversion, the retinal image, or proximal stimulus, is inverted and upside down. Here, light from the superior visual field falls on the bottom of the retina, and light from the inferior visual field falls on the top parts of the retina. Additionally, because of this inversion, light from the contralateral visual field, that is, the visual field opposite to the viewing eye, falls onto the temporal retina, while the ipsilateral, that is, the visual field on the same side of the viewing eye, falls onto the nasal retina. At the retina, the proximal image is coded by rod and cone photoreceptors, and neurons in the retina, particularly retinal ganglion cells, which work to convert light information into electrical signals and transmit these signals to the brain. Retinal ganglion cells can be large and referred to as parasol cells, or small and often referred to as midget cells. Parasol cells are mainly located at peripheral locations and pull information from rod photoreceptors, while midget cells are found primarily at central regions at the fovea and synapse mainly with cone photoreceptors. The axons of retinal ganglion cells all leave the eye at one common point, the optic disc, and the neuronal bundles of fibres form the optic nerve. If we look at the optic nerve, a cross-sectional view, we see that the arrangement of axons is orderly such that the spatial position of retinal ganglion cells in the retina is preserved. This is because adjacent neurons in the retina project together to adjacent locations in the optic nerve, and this order is maintained all the way to the cortex of the brain. This results in the formation of a retinotopic map, which preserves the spatial layout of the visual scene in the brain, such as at the striate cortex. Visual information leaves the eyes via the optic nerves and then come together again to form the optic chiasm. At the optic chiasm, nerve fibres undergo a process of decausation, or crossing over. But note that only 50% of the fibres cross over in a process called semi-decausation. The crossing over is highly organised, with only fibres from the nasal retina and ipsilateral visual field crossing over. Temporal fibres, those carrying information from the contralateral retina and visual field, do not cross over, but remain on the same side. This semi-decausation ensures that afterwards, visual information received by the 2 hemispheres in the brain is from the contralateral or opposite visual field, but most importantly, the same contralateral view from both left and right eyes. Following the optic chiasm, fibres come together again, but still preserving their retinotopic arrangement to form the optic tracts, which then project to the lateral geniculate nucleus, or LGN, of the thalamus. Lateral, which represents its location on the thalamus, and geniculate refers to its shape, which looks like a knee, which in Latin is genu. The lateral geniculate nucleus is an important neural structure in relaying information to the brain. If we look at the cellular organisation of the lateral geniculate nucleus, which is evident in a cross-sectional view of it, 6 distinct cellular layers are evident. The first 2 layers are referred to as magnocellular layers and made up of large magnocellular cells, while the following 4 layers comprise of smaller cells and are referred to as parvocellular cells, after parvus for small. If we trace back to the type of retinal ganglion cells that end up projecting to magnocellular and parvocellular layers, we will see that magnocellular cells receive their inputs from the larger paracel cells in the retina, while parvocellular cells receive their inputs mainly from smaller midget cells in the retina. Now, in between the 6 layers, another class of cells exists, which are often referred to as coniocellular cells, and they receive inputs from midget cells, particularly those that selectively code short wavelength information. If we look further at the organisation of the lateral geniculate nucleus, we see that the retinotopic map is preserved across all 6 layers, but inputs from left and right eyes are kept separate and connect exclusively to different layers, particularly ipsilateral fibres, that is, from the same side, synapse with layers 2 and 3 and 5, while contralateral fibres, that is, from the opposite side, synapse with layers 1, 4 and 6 of the lateral geniculate nucleus. From the lateral geniculate nucleus, neuronal axons then project posteriorly through the internal capsule of the brain to the cortex through optic radiations to synapse with the striate cortex, or primary visual cortex, which is the first part of the brain to receive and process visual information. Note that approximately 80% of the fibres from the optic tract go to the lateral geniculate nucleus. 20% of the fibres bypass the lateral geniculate nucleus and synapse with another visual structure, the superior colliculus, which is located on the tectum of the brainstem. The superior colliculus is an important structure in the control of visual orientation, particularly in eye movement control and saccadic control, and is another pathway into the visual cortex. In summary, the prestriate areas of the visual system is highly organised to detect and transmit light information to the brain. Notable features of the prestriate areas are the preservation of the spatial layout of the visual field in terms of retinotopic map, and semi-decausation of projections to ensure that each cortical hemisphere processes the contralateral visual field with signals from both left and right eyes.